Hello everyone, my name is Claudia Gentile and I'm a principal scientist at AGC Biologics. Today I will give you an overview of AGC expertise in cell therapy from early development through to GMP manufacturing and patient supply. To briefly introduce myself, I have 5 years of experience in process development and manufacturing for cell and gene therapies. I started my career in cancer research after completing my master's degree in biology applied to biomedical research at the University of Milan. After this experience, I joined AGC Biologics, where I combined scientific expertise with process development activities, contributing to scalable manufacturing strategies for advanced therapy. Now let's talk about AGC Biologics. We are a global company with a network of 6 sites across 3 continents, the USA, Europe, and Asia. We operate in Seattle for North America, in Europe through Copenhagen, Heidelberg, and Milan, and in Asia through Chiba and the most recent site, Yokohama. This global footprint, supported by more than 2,800 employees, allows us to serve over 350 customers worldwide. Each site has specialized capabilities from mammalian and microbial production to viral vectors, plasmidic DNA, and of course cell therapy, which is a key focus of the Milan site. Our experience is supported by a robust track record. In protein-based biologics, we have brought several commercial products to market and have worked on a wide range of products. But most relevant to our discussion today is our success in cell and gene therapies. We have been a part of bringing 11 commercial viral vectors and 7 commercial cell therapies to market. We have manufactured over 400 GMP batches of viral vectors and have treated over 550 patients with autologous cell therapies. This extensive experience supplying clinical trials in both Europe and the US demonstrates our deep commitment and capability in this advanced field. Focusing on the Milan facility, this site is a cornerstone of our cell and gene therapy services, with 30 years of industry-leading expertise and 10 commercial approvals. The Milan site is approved by major regulatory bodies, including the EMA, the FDA, and FDA. We offer both clinical and commercial manufacturing for cell therapy and viral vectors with multiple GMP rooms. Our capabilities include a wide range of technologies, from adherent and suspension bioreactors for viral vectors production to over 10 dedicated suites for both autologous and allogeneic cell therapies. We can handle the main primary cell types, such as hematopoietic stem cells, T cells, NK cells, and mesenchymal stem cells, and more. A key strength is our in-house analytical platform with more than 160 tests available on site, ensuring fast and robust quality control. Our cell therapy expertise is built on several key pillars. First, a strong experience in process development, including transfer and internalization of both autologous and allogeneic process from our clients. Second, extensive hands-on experience with the most common primary cell types, as previously introduced. And third, full support and customization, development and validation of both manufacturing and analytical methods. All of this is supported by a fully GMP-compliant facility, ready for both clinical and commercial supply, aligned with EMA and FDA guidelines. This timeline showcases our extensive history of process validation at the Milan site over the last decade. We have successfully completed 90 process validation campaigns, 6 for cell products and 13 for vector products. As you can see, our work has spanned various product types, including T cells, hematopoietic stem cells, and different viral vectors, like lentiviruses and retroviruses. This continuous activity demonstrates our deep experience and readiness for late-stage and commercial manufacturing. And our pipeline for validation remains strong, with more programs already in preparation for the coming years. This slide shows the landscape of the ex vivo commercially approved cell and gene therapy products. The products highlighted in green are those manufactured by AGC Biologics, including, for example, Strimvelis, Zalmoxis, Libmeldi and Lebmeldi, and Woskiria. These examples underscore our ability to navigate the complex journey from development to market approval. Now, let's dive deeper into our cell therapy capabilities. This diagram illustrates step by step all main phases of a cell therapy manufacturing process. Obviously, depending on the specific process, some of these steps may be skipped. In detail, a cell process starts with the collection of starting material and isolation or enrichment of specific cell population. Following this, cells are stimulated to grow with the proper cytokine cocktail or activator. In this context, we have extensive experience handling both fresh and frozen starting materials, and we are experts in both open and closed processes, using a wide range of equipment for cell selection. Then, if required, genetic modification can be carried out using viral vectors such as RVV and LVV, or through gene editing techniques. In particular, gene addition using viral vectors represents one of our key areas of expertise and core strengths. The cells are then expanded to reach the final target concentration, yielding the drug substance, which is then formulated into the final drug product, ready for the patient. And what is our main role in this process? The process development team is the first point of contact for our clients, acting as a GMP-like environment to de-risk the process before it's transferred into the official GMP manufacturing suite, ensuring a smooth and efficient transition. Specifically, our strength lies in offering customer maximum flexibility thanks to our broad expertise, which enables us to easily adapt to a wide range of specific requirements. In fact, to support these complex processes, we proactively test and acquire the most advanced automated systems for every step of manufacturing, from isolation and gene transfer to expansion and cryopreservation. This slide shows just a sample of the cutting-edge equipment we use, like the Climax Prodigy for an all-in-one method or the combination of different instruments for a modular approach. By having different toolboxes of automated closed systems, we can design a bespoke modular workflow that is perfectly tailored to the specific needs of each client's product. Let's look at a specific example, our hematopoietic stem cell platform. On the left, you see the traditional open system, which involves many manual steps. The open system represents the historical procedure used for this process, in which we have developed extensive experience. Moreover, this procedure has been internally optimized to eliminate the use of adjuvants while still achieving high transaction efficiency, thanks to modifications in certain process parameters and reagents. On the right is our goal, a fully closed and automated platform. This system integrates the initial CD34 selection using the LP enrichment program with the subsequent transduction and cell culture steps managed by the HSCE program. Even the fill and finish stage can be fully closed and automated with automated instruments. For example, for this final step, we worked internally to optimize the procedure using as instruments the Q. We have gained experience with this closed and automated system by working closely with clients to implement several optimizations tailored to the requirements of the specific process. The shift from an open manual system to a closed one is critical for assuring consistency, scalability, and the highest level of safety and quality. We applied the same approach to our T-cell processes. Our experience covers the full spectrum, from fully open manual systems to modular workflows, combining different instruments for each step, all the way to fully closed and automated manufacturing using the Climax Prodigy TCT program. In this field as well, development initially began with an open process, and extensive experience accumulated over time enabled the successful commercialization of products such as Zalmoxis, that I have mentioned before. Since then, much of our work has focused on optimizing and adapting standard programs to fit our client-specific processes by integrating all manufacturing steps through a broad range of tools, enabling the use of frozen starting material and supporting future applications. This allows us to support clients at every stage of their development phase, while ensuring a high degree of process adaptability. For allogenic therapies, the requirements are a little bit different, particularly in terms of scale. The main challenges for this setting was evaluating systems for large-scale cell expansion and high-volume fill and finish operations. Our development work addresses these requirements for T cells and specific T cell subpopulations, such as NK cells and Tregs. The process includes immunomagnetic selection and/or depletion of a specific population using Climax Plus or Climax Prodigy instruments, an optional gene transfer step, and a large-scale expansion phase. For the initial selection phase, we leveraged the extensive experience gained from previously described autologous processes. And however, the core of our work has primarily focused on the cell expansion phase. To this end, we developed scalable expansion protocols using two main platforms: G-Rex systems, capable of generating up to 20 billion cells in just 10 days, and bioreactor systems, such as Xuuri, for even larger scale production. Developing this expertise is crucial for the commercial viability of allogenic therapies. More recently, our development expertise has also extended to other primary cell types, like mesenchymal stem cells. In this case as well, the same approach was applied, including an initial development phase in an open system involving direct plating for MSC isolation, and at the same time, the evaluation of different 2D culture conditions to define a cell expansion protocol. Subsequently, several improvements were introduced to progressively close the system and to move toward larger scale 2D culture system through the use of cell factories. Finally, for GMP compliant large scale production, a fully closed and automated system was developed for both the isolation and the expansion of these cells using more recent technologies, such as the hollow fiber bioreactors. In parallel, in a highly innovative way, We have also developed specific engineering techniques through LVV transduction, first in 2D systems such as wells and flasks, and then in 3D systems using different platforms such as CEPAX and Terumo bioreactor. Overall, this new expertise has enabled the development of different types of protocols for a variety of applications, including, of course, cell therapy, but also the exosomes production. The wide variety of processes I have just described would not be possible without adequate infrastructure. Our facility is specifically designed to support this process flexibility, providing different GMP environments tailored to the specific needs of each product. We have traditional dedicated GMP rooms, ideal for autologous open processes. We also have the ballroom concept, a large open plant clean room, ideal for producing a large number of batches where autologous hematopoietic stem cells and T-cell process are completely closed and automated. Furthermore, we are moving towards the dense floor concept, an advanced evolution where highly automated closed and mobile platforms operate within a shared GMP space. This innovative approach maximizes flexibility, reduces complexity, and enables truly scalable manufacturing, so it fits perfectly for the allogenic processes. Of course, manufacturing is only a part of the story. A robust analytical platform is critical for ensuring product quality, safety, and potency. Our platform covers all necessary testing for both product release and characterization, including tests for safety, purity, and potency. As shown in this slide, some of these are platform-specific, while others are unique and developed specifically for each product. Having this comprehensive analytical capability in-house is a key part of our integrated solution. So, to summarize the key points, AGC Biologics brings extensive cross-functional experience across multiple fields, supported by a strong track record of successful product launches and a broad portfolio of development and manufactured therapies. In cell and gene therapy specifically, we have contributed to 11 commercial viral vector products and 7 commercial cell therapies, with extensive manufacturing experience encompassing more than 400 GMP viral vector batches and the treatment of over 550 patients with autologous cell therapies. I would like to highlight 4 main aspects. First, our key strength lies in deep technical expertise combined with continuous innovation. We work across a wide range of cell types and consistently integrate cutting-edge technologies into our processes. Second, we offer manufacturing flexibility supported by a state-of-the-art infrastructure, ranging from dedicated GMP suites to advanced dense floor concepts designed for highly scalable production. Third, we provide end-to-end integrated solutions, acting as a single source partner covering the full value chain, from viral vector manufacturing and process development to analytical testing. Finally, we believe in a true 360-degree client partnership, supporting our clients at every stage of their journey, from early development through to commercial launch and beyond. This brings me to the end of my presentation. I hope this has given you a valuable insight into AGC Biologics cell therapy expertise. Feel free to contact me directly with any questions. Thank you very much for your attention.